Hello, everyone. Welcome to another part of the new speaker series stream. And I'd like to introduce everyone Dr. Elvin Chizenga, who is a certified biomedical physicist with a PhD in biomedical sciences, specializing in biomedical optics and biophotonics. He's a founder and CEO of Lightaceutics, a startup developing light therapies and optical technology for healthcare. His current research focuses on photobiology, electrophysiology, and bioelectromagnetism applications, with multidisciplinary expertise spanning biology, biochemistry, biophysics, and AI tools. Elvin is committed to closing this knowledge gap through his podcast, Dr. Elvin C, Light Up Media, and is developing medical innovations through Lightaceutics. And so I'll leave it up to you now.
All right. Thanks for the introduction. So do we have to share the slides? Okay, maybe let me download it so that we don't share the entire tab. I can just do present. Okay, there we go. Right. Yeah. So I'm just gonna go quickly through the slides that I have today. This is a very broad topic, so it's kind of tricky to actually go about it entirely. So I actually spoke to Dr. Rajiv already that today I'm just going to do a very brief introduction as to what we do when it comes to photobiomodulation and photodynamic therapy. But our agreement is that later on, I am going to come a couple of more times to go into the details of the topics that I want us to get an introduction of today. So
I'll just keep talking.
I just want to give you a bit of a perspective as to what really is light therapies. You see, these terms can sound fancy: photodynamic therapy, photobiomodulation, but really they are very basic terms. What they mean in photobiomodulation, you're just looking at the interaction of light with biological tissue, when in fact you're using the photons of light to cause tissue regeneration. And in photodynamic therapy, that's just another different type of process where you're using a different technique that employs the same thing, which is light in this case, but uses other molecules to cause phototoxicity that is used for the treatment of different types of diseases. So I am going to continue. Are we still sharing the slides?
Somebody was just asking a brief introduction from where you studied, which college, and a little bit about yourself.
Oh, okay. So I started off my education in Malawi. So I did a diploma in biomedical sciences at Malawi Adventist University. And then after that, I think I only worked for about 2 years as a laboratory technician. And then I moved to South Africa, where I joined the University of Johannesburg. I did my master's in biomedical technology, and then I continued with my PhD in biomedical sciences. So all my postgraduate research was really on photodynamic therapy, which is the use of light for the treatment of different types of diseases that I'm going to talk about. But essentially, after I completed my PhD, I did a very brief postdoctoral fellowship before I founded my company. And then my company is only less than a year old. So I'm still a scientist at heart and an entrepreneur because of the company that I'm actually building right now. So basically, that's the most brief introduction I can give. Is there anything else I need to clarify? All right, so I will go right into the presentation. I'm just going to skip through the slides because I want us to just have a good introduction and then have a lot of questions that we can spend much time on to discuss. So I just want to give you a bit of a perspective as to why this conversation is important. Okay, the slides I'm sharing, I cannot seem to skip. I think it's locked.
You should be able to go to the presenter window.
Okay.
Right now.
All right. So thanks for that. So I just want to give you a bit of a perspective as to why this topic is very important. So when we're talking about light as a therapy, we're going back in history where the first observations of light working, the observations of light having their biological effect on humans was observed back in the Egyptian civilization when people observed that sunlight had a lot of biological effect on not just human beings, but on plants and on the treatment of different types of diseases. So this brought about the origins of laser sciences, which we really can trace back to the 20th century. That's the earliest writings that we can find about the principles of light in medicine. So laser therapy was later on discovered as an actual treatment procedure early in the 1960s by scientists that paid much attention to now having observed the biological effects. Now people wanted to understand, okay, these biological effects we can see, but how does this happen? So the actual science of using light as a therapy, we would say that was discovered in about 1960s. So this guy actually demonstrated the first time using an H&E laser that could stimulate wound healing in mice. So I'll talk a little bit about the wound healing process, but the first observation of the biological effects of light were observed using how this laser stimulated wound healing in laboratory mice. So at present, light, you'll see that I'm talking about lasers a lot because that's the type of light that we use in medicine. But any type of light, including natural light from the sun, even the light that we have in our rooms, the tungstens, the LEDs, all these forms of light have got some biological effect. Some that's negligible, some that's actually very important that we need to also discuss. But when we talk about the medical applications of light, we're really talking about lasers. All right. So lasers have become an essential tool when it comes to the medical applications of light, and that is where we are going to pay most attention. So I just want to talk about the electromagnetic spectrum. I don't want to go into the technicalities of what this image means, but I just want to point out that the electromagnetic spectrum is a very broad spectrum of electromagnetic radiation. So what we refer to as light is usually a very tiny portion of the electromagnetic spectrum. So our eyes can only comprehend light from about 420 nanometers, which is that rainbow color on the picture that you can see there, until somewhere about 780 nanometers of wavelength. But that doesn't mean that that's the only radiation that we have on the electromagnetic spectrum. This is a very wide electromagnetic spectrum. In fact, we can only measure it to certain limits, but that doesn't even mean that that's the end of the spectrum. It only means that that's the end of how much we can measure the electromagnetic spectrum. So if you start from high to low, you will see that there's non-ionizing radiation from the highest wavelength to ionizing radiation from the lowest wavelengths of light. So this is very important because when we're talking about light, we're talking about three characteristics of light. We're talking about the wavelength of light. So when you're talking about the wavelength of light, that is when I refer to the wavelength between 420 nanometers to 780 nanometers, which is the visible spectrum of light that we can see. But beyond 780 nanometers, you're moving into infrared radiation, which is light we cannot see, but you can see it when you're using, let's say, night vision glasses. When you're using infrared glasses that actually filter out certain wavelengths, you can be able to see infrared light. And then from the infrared light, you go into the microwaves. These are the same microwaves that we use for heating our foods. And then you go higher into the radio waves. So these are the signals that we receive, our Wi-Fi signals that we receive our radio transmissions from. So the higher you go in the electromagnetic spectrum, the less energy of the electromagnetic spectrum that you will receive. That is why when you talk about higher wavelengths of light, you're talking about very low energies that we refer to as non-ionizing radiation. On the opposite side of the electromagnetic spectrum, from the visible spectrum below 420 nanometers, that's when you start to see the ultraviolet light from the sun. So the ultraviolet light from the sun is also light that we cannot see with our eyes, not because it's not there, just because our eyes cannot really comprehend that wavelength of light. So we're going down from the ultraviolet light into the X-ray. We use X-rays for medical imaging. We use X-rays for many other things, even in industrial applications that are not medical. They use a lot of X-rays to image different types of materials. And then there's gamma rays. Gamma rays are the furthest from the down the electromagnetic spectrum, and these are very high energy wavelengths. So remember, I keep repeating the fact that the lower you go down the electromagnetic spectrum in nanometers, the higher the energy becomes. This is very important because there's a very difference between ionizing radiation, which is wavelengths below the visible spectrum, from the ultraviolet to the X-ray to the gamma rays. That is why we talk about gamma rays as toxic to biological tissue. Why? Because they have so much energy that is ionizing in nature. Even ultraviolet light is below the visible spectrum. It is not too much energetic like the gamma rays, but it is also energetic. That is why if you have too much sun exposure, you can get sunburned, and sometimes you can actually be predisposed to things like melanomas. So remember, if you forget anything, just always remember that when you're going down the spectrum, from the visible wavelength lower down the spectrum, you're looking at high energy electromagnetic radiation. If you're going up the spectrum, you're looking at low energy electromagnetic spectrum. So what are the clinical applications of light? So when I talk about the clinical applications of light, usually we're talking about the visible spectrum. And remember, when we're talking about light, we're talking about the part of the electromagnetic spectrum that you can see. But if you sit back and talk physics, light could be anything from the things we can see to the visible spectrum that we see. So in the clinical sciences, in medicine, in healthcare, what we usually use for therapy is the visible spectrum of light, starting from the near infrared region, going down into the blue light that gets merged into the ultraviolet. So the reason why this is done is because we have observed clearly the beneficial effects of this part of the spectrum, and also the toxic effects, because light has got a biological effect. It only means that it can interact with biological tissue. Whether that interaction results in a positive outcome or a negative outcome depends on many things. Depends on the wavelength of light, depends on the type of tissue that's affected, and it depends on how you use the light in the physics part of it, the power output, the irradiance, the fluence of the light that you used. All these are very important aspects of the light that would determine whether the light is either toxic, beneficial, or just neutral. So one of the most common applications of light is PBM, which is photobiomodulation. So photobiomodulation was formerly known as low-level laser therapy, but now we call it photobiomodulation. And the term photobiomodulation only means that you're using photo, which stands for light. So photobiomodulation, bio for biological tissue, modulation, simple English, you're modulating a process. So photobiomodulation is a combination of three words. You're using photons of light to modulate biological tissue. So usually in photobiomodulation, we use red light or near infrared light. Near infrared light. Remember, I mentioned that the biological outcome when light is exposed to a particular tissue will depend on many factors, including the wavelength of light used and the type of biological tissue exposed. So the reason why we use red light for photobiomodulation is because red light has got just the right amount of energy in terms of wavelengths that biological tissue can interact with in many aspects. So there's a lot of ways in which tissues respond to red light when exposed to biological tissue. So there's a lot of research that has been done that when light, oh, the slides are not moving. Okay, I've changed here. I'm on my fourth slide.
I can help.
Please come help me. It means that it's not moving.
So maybe you can just summarize quickly through those which did not move, like bring the audience back.
Okay, is it changing now there?
Yeah, so now maybe.
Okay. So
Very quickly, yeah. Most of the people have been listening, but it's nice.
So I was really talking about the electromagnetic spectrum, and you guys were not seeing the whole thing. Oh man, should I go back to that?
Yeah, like summarize. You can, why not? Yeah.
Oh, I apologize about that. I was seeing my slides here, and I see that you guys were not really seeing what I was looking at. So I'm just gonna go back. I spoke about the electromagnetic spectrum and spent a lot of time talking about it. What I really meant here was that if you look at the electromagnetic spectrum, that picture, the only part of light that our eyes can observe and our brain can actually process and see is light from 420 to 780. So on that picture, you're looking at the part that looks like a rainbow from red going down to blue into ultraviolet light. So that's the only part of light that our eyes can actually comprehend. But if you go up the spectrum into the infrared, microwave, and radio waves, that still is electromagnetic spectrum energy. That's still a lot of wavelengths that our eyes cannot see, but they are there. If you go down the spectrum, it's the same thing. You're looking at light below the visible spectrum. This is high energy, lower wavelengths electromagnetic radiation. So into the ultraviolet light, into the X-rays, into the gamma rays. So this is what I meant when I say that the electromagnetic spectrum is a very broad range of light energy. This is irradiation. So the fact that the only part of the spectrum that we can see is the visible spectrum does not mean that the other ones are not relevant. They are always relevant. We just can't see them. All right. So that's what I really meant by that. And then I was now talking about the clinical applications of photobiomodulation. So there's a lot of applications that we can actually benefit from photobiomodulation, but I kept saying that we are looking specifically at red light. And the reason why we're looking specifically at red light is really because biological tissue can interact with red light and respond by uh, producing different types of signaling molecules. So there's a lot of things that red light can do. It can actually make cells to produce antioxidants. Red light can actually make cells to produce angiogenic factors. So angiogenic factors, these are molecules that your body needs to make new blood cells. That is why when you photobiomodulate tissue, when you expose red light to tissue, that particular tissue, if it's a wound, it would be able to produce angiogenic factors. These are molecules that are important for the production of new blood vessels. So red light can also produce a lot of growth factors in the extracellular matrix. So when you're looking at, let's say there's a wound and this wound takes time to heal, there's a very much clinical area of focus, which is photobiomodulation for treatment of diabetic wounds. You've heard about diabetic wounds and the fact that they cannot heal properly. That is why when a diabetic patient has got a foot ulcer, for instance, it keeps getting worse until the time that the foot has to be amputated. But we can use photobiomodulation to actually reverse the process. And how red light reverses that process is really by producing growth factors in the wound that a diabetic patient cannot normally produce. So these are all the things that red light can do. And the reason why red light is able to do all these things is because the photons of light in the red region of the electromagnetic spectrum are absorbed by our tissues. And when our tissues absorb those photons of light, they get activated in different ways by producing the growth factors, by producing angiogenic factors. They release antioxidants and many other things as well. So the other thing that I wanted to talk about is the issue of photodynamic therapy. So photodynamic therapy is different from photobiomodulation in that photodynamic therapy is another technique that, instead of using it to cause tissue regeneration, to produce a positive effect in the tissue, you're actually using it to produce the opposite effect, which is toxicity to the tissue. Why do we need to produce toxicity in tissues? A very good example is when we want to treat cancer. We know that cancer is the overgrowth of cells, and there's a lot of treatment procedures that we have. We have chemotherapy, we have radiation, we have newer therapies coming up. But the idea, when treating cancer, you want to remove the entire tumor. So here, instead of using chemo, radiation, and other forms of therapy, we want to use light to remove the tumor. So we are using light, and this is the features of light that are toxic to biological tissue. I just explained that light is beneficial. How then does it become toxic to biological tissue? So in photodynamic therapy, you employ a third component in the therapeutic process, where in photobiomodulation, you're just using red light to expose the photons in the red light energy to biological tissue. In photodynamic therapy, you are using different types of light. It can be the red light, it can be blue light, it can be green light, but you add a second feature, and this feature is a molecule called a photosensitizer. So a photosensitizer is basically a dye molecule that will absorb the photons of light. And when it absorbs the photon of light, it becomes activated. So in its activated state, it is the photosensitizer now that will react with molecular oxygen in the tissue, and then it will cause reactive oxygen species and free radicals. So in photodynamic therapy, this is what we really mean, that instead of just using light to photobiomodulate the tissue, we are actually using a toxic compound that will absorb the light. It becomes toxic enough to actually cause tissue toxicity. So that is how we are only able to use it for treatment of cancer, and we can also use it for treatment of infections. So there's a lot of bacteria, for instance, that naturally produce these light activatable photosensitizers in them. A good example is the bacterium that causes acne, which is called Propionibacterium acnes. This bacterium actually produces a photosensitizer in it that gets activated by blue light. So you've seen on Amazon, on eBay, you can actually get a face mask that's blue in color that people say is used to treat acne. And you wonder, how does this treat acne? The reason why it treats acne is because the bacterium that is involved in the whole pathogenesis of acne produces naturally a photosensitizer that can get activated by the blue light that's on the mask, excuse me, and it can actually cause the removal of the bacteria, and hence it can help in the treatment of acne. But there's a trick here. Remember, I'm talking about blue light. So there's other people that have tried to use red light for the treatment of acne, and that can be dangerous, very dangerous, because remember, acne is the overgrowth of the overproliferation of follicular cells under the skin that produce hair. All right. So that overproliferation of cells produces a lot of sebum, a lot of oils, a lot of debris. That is the basis of acne. Now, if you're going to use red light on a red light mask, assuming that you want to treat acne, what you're actually doing is you're making the condition worse, because instead of using the right light energy, you are using the wrong light energy. Instead of using blue light, which is meant for treatment of acne, you may not be aware, you just take any random facial mask and you use red light for the treatment of acne, and you just make the condition worse, the condition even worse. That's exactly what I meant by the biological effect. The outcome really depends on the type of light that you're going to use, and this is very important. This is something that you always need to be aware of. So
thank you, and I think we have some. While you get a pause, we have some comments for you.
Okay.
I can read it for you, and you can also read it here.
All right.
Right. So the first one is from Avery Bennett. He has mentioned excited to learn about the applications of photonics in medicine. So that's the wonderful start to your topic. We have Exploring Latitude, which is saying great topic with a thumbs up. So wonderful. And Dominique has said interesting presentation. Avery says when it comes to different types of electromagnetic waves, what makes some safe to be exposed to and some dangerous? And then I'll take one more question, and then you can take later. So Rab Azizi says, did not know about this before. Glad I joined today. So maybe you can take Avery's question, and then we'll come back to some other questions.
Okay, so the question is,
when it comes to different types of electromagnetic waves, what makes some safe to be exposed to and some dangerous?
Okay, so what makes other forms of electromagnetic radiation safe and some dangerous is really the type, the region on the electromagnetic radiation that we're talking about. Remember the picture that I showed you. So you want me to be here?
Yeah.
Okay, so remember the picture that I showed you of the electromagnetic radiation, electromagnetic spectrum. That picture explains the different energies that the light has. So let me quickly go back to that picture because it will help answer the question.
Our audience are very attentive and they have some great questions coming your way.
All right. So let me just point out to this picture. Can you see the picture?
Yes.
All right. So this is what I meant. When you're looking at the electromagnetic spectrum, you're looking at a very broad range of light. And remember, I keep saying light as the entire electromagnetic spectrum, when in our English terminology of light, we're talking about the visible spectrum. But in physics, we're talking about the entire electromagnetic spectrum. So the trick about the electromagnetic spectrum is that the fact that what we can only observe is the visible spectrum, it doesn't mean that all the other types of electromagnetic radiation that we cannot see are not relevant. So that is the reason why, for instance, for radiotherapy, to use radiotherapy for the treatment of cancer, you are actually using some high energy ionizing radiation, which are in the gamma region, that are toxic to tissue. And the reason why they are toxic to tissue is because they are inherently high energy radiation. If you're going up the spectrum above the visible electromagnetic spectrum, you're looking at infrared. The remote that we use to change a TV channel, that's infrared energy. The reason why it doesn't have a direct effect on biological tissue really is because it does not have that much ionizing energy in it. When we heat up food in the microwave, for instance, the only thing that the microwave does is to cause the increased vibration of water molecules that end up heating up the entire food or get the entire food cooked. And that's another important thing. There's people I've heard talk about the dangers of using microwaves, the dangers of eating food that's cooked or warmed up in a microwave. And there's other wrong information out there that people would say that food that's cooked in a microwave would cause cancer. That is wrong information, because microwaves are not, they are non-ionizing radiation. They do not ionize biological tissue. What they do, they just cause a lot of vibrations that end up heating up the tissue. So the dangers of eating microwave food is not because cause cancer or anything, but really it's because when you are eating food that's been heated up in the microwave, the microwave kind of destroys most of the nutrients in the food, because remember, for a nutrient to be what it is, it depends on the conformational structure of that nutrient. So if you use microwaves just randomly break the bonds by overheating the food, so you end up destroying the nutrients. That's why you'll be eating food as substance, but you're not benefiting from the nutritional value of that food, because the microwave actually breaks all those bonds to heat up the food, and you may not have enough nutrients in the food. So that's really the most important thing. So to summarize the question that you asked, the electromagnetic spectrum from below the visible spectrum to going up the visible spectrum, you're looking at the different energies of light, and the lower you go, the higher the energy. The higher you go in the spectrum, the lower the energy. That is why you really need to be very careful with light that you're exposed to going down the visible spectrum, starting from the UV region. The sun produces very good amount of energy. We make vitamin D because of our exposure to the sun. Our skins, our melanocytes produce melanin. People with darker skin like myself, the more I'm exposed to the sun, the more melanin I'm going to produce. And melanin is a very important compound in our bodies, not just for skin color. So this is very important because light is very beneficial, but when you're going down the spectrum, it becomes more ionizing, and it can injure biological tissue. So basically, that's all about that. Was there a second question before I?
Yeah, I mean, there's a lot of questions.
There's a lot of questions, so I have to change tabs to see the questions. Do you want me to just read them?
Yeah, I can read some of them for you.
All right.
What made you first interested in photonics?
What made me interested in photonics? Really, I was interested in cancer biology, not photonics. But when I entered my master's program to study cancer biology and cancer therapy, the type of therapy that I started with was photodynamic therapy. So when I ended up doing photodynamic therapy, observing the beneficial effects of light in the lab, seeing what light does to tissue, because my master's and my PhD studies really focused on experimental work where I would culture cells in vitro and then expose those cells to light and then see how they change, see how they grow, see how they get destroyed. So I started observing these things, and I got very intrigued with the role of light on biological tissue. You can literally see when you expose light to biological tissue how these cells can either just grow as fast, or how this light can actually cause phototoxicity to the cells. So I got so much interest into looking at the actual mechanisms that actually take place for this to happen. And when I started studying deeper into the field, I ended up learning a lot of intriguing things about bioelectricity, bioelectromagnetism, and now my research focus really is looking at bioelectricity and bioelectromagnetism, because I have learned that as people, we are essentially electromagnetic beings. I think our medical sciences were built on a biochemical model of education. So we perceive the body to be a biochemical machine. Like when we talk medicine, we're talking about biochemistry. But when you now come back to look at the biological effects of light and how our bodies respond to light, we actually generate some type of electromagnetic radiation ourselves. We can produce infrared radiation. That is why we can produce heat. We can share heat, because that is the amount of electromagnetic radiation that we can produce. So there's a lot of information. There's the science of our energies, for instance, how we can interact with people, with different people, just by tapping into our energies. So this is a very, very interesting field of study. And why I'm very interested about it is because I'm learning as I go, as I get into the research, that I think we need some kind of a paradigm shift in how we understand physiology. Instead of looking at the body as a biochemical machine, our bodies are more complex than that. We are essentially bioelectric beings as well, and we can interact with different types of light. We can interact with electricity in many ways, and this is very important. So that's really why I have so much interest in this field. And let's get more questions. How does infrared light stimulate cells? Okay, so infrared light does not directly stimulate cells. Remember, infrared light, you're looking at light that's emerging from the visible spectrum up the spectrum, and it's light that we cannot even see. So that is the reason why I gave an example of a remote control that uses infrared energy. So infrared light does not directly stimulate biological tissue. However, infrared light has got a biological effect, which is photothermal. Photothermal meaning that it can heat up tissue. So there's a very big difference between stimulating tissue, because when you talk about stimulating tissue, you're talking about photobiomodulating the tissue. But when you're talking about other effects like the photothermal effects, that's not really tissue stimulation. That's just the advent effect of exposing light that causes vibrations in the tissue that heats up the tissue. In the same way, upper energies like the microwave can literally cook up tissue. So starting from the infrared going up, you're looking at a type of energy that heats up tissue. Even red light that's so close to the infrared region. Red light can also cause hyperthermal effects as well, because the higher you go, the lower energy the light is, but it's more likely to heat up the tissue because that's the type of energy that it has. So I hope I answered that question. So infrared won't stimulate your cells directly, but it can cause some form of thermal effect. In physiology, we also use infrared light lasers in our treatment. Yes, so in physiotherapy, you use infrared light for the same reason. So remember, when you when you cause a bit of photothermal effect, you're actually increasing some, how should I put it? You're actually also increasing some cellular processes, not by photobiomodulatory effect in the same way red light would, but it would also cause some biological effect based on the light energy that's being delivered. So remember, the most important thing is the light energy that's being delivered to the particular tissue. So yes, infrared light has been used. In fact, infrared light is used for many other applications as well, including in laser aesthetics. People are using a lot of infrared light as well instead of visible light, because infrared light has got more energy. And remember, energy is not only ionizing energy. I'm talking about energy in the terms of the higher the electromagnetic spectrum you're going, the higher the wavelength, though the lower the energy when it comes to ionizing radiation. So how does blue light therapy work on acne? So I mentioned that in acne, blue light therapy works because acne can be caused by a bacterium called Propionibacterium acnes that produces a compound in it that is phototoxic when exposed to blue light. But you should remember that not all acne is caused by this bacterium. There's other acnes. I think there's different types of acne. I just found out that. But there's times when there's acne without the presence of the bacterium. So the only time that a blue light face mask would work on acne is when there is active proliferation of this particular bacterium. If you're looking at a type of acne that doesn't have the bacterium, Propionibacterium acnes in it, then blue light won't be beneficial because there won't be any type of photosensitizer to cause the phototoxicity. That is why I even gave an example of people that have mistakenly used red light facial masks for the treatment of acne. This is very dangerous because there's a difference between acne as a lesion and acne as a scar. Because when you're looking at an acne scar, you're looking at an acne lesion that has started forming scar tissue because of collagen deposition that is produced by hyperproliferation of cells. So red light stimulates cells to grow even faster. So someone would have an acne scars because already they have cells that are growing fast to cause an acne scar. So if you mistakenly use red light, assuming that you're using it for the treatment of acne, you're literally just making your acne scars even worse, because now what will happen is that the red light will even hyperproliferate the cells that are already hyperproliferating. So I hope that makes sense. How long does a normal session usually take? A normal session for the therapies or the presentation? The therapy. So there's different types of therapies. I don't know in what context you're asking about the session, but for instance, if you're looking at my research in the treatment of cancer using photodynamic therapy, what you'd have to do is you have to optimize the amount of time it would take for you to actually cause a noticeable effect. And remember, if you overdo it, you can actually cause photothermal effects that are not something that you want. If you don't do it at the right amount of exposure time, you won't have the actual effect, because the transition of the activation of this photosensitizer is also time dependent, and of course light energy dependent as well. So the amount of session really depends on the procedure that you're doing. If you're doing a procedure like let's say you're doing a procedure for a facial procedure to remove wrinkles, for instance. There's people that are using procedures to remove wrinkles. So if you're doing a procedure like that, you have to optimize a lot of things. You have to optimize the type of skin the person has. You have to optimize the type of light that you're going to use. If you're going to use lasers in the infrared region, then that's another type of optimization. If you're going to use red light or green light, you'd have to always optimize the amount of time you need per procedure. And it's not just a standard procedure, it also depends on the different types of patients that you're going to have as well. Remember, there's other people that are darker than others. There's other wrinkles that are tougher than others. So it really depends on the type of procedure that you're doing. So if maybe you could ask the question again as to how long does a normal session take? What session are you referring to? But if I would answer it broadly in general, I would say that the session really depends on what it is that you're doing and what it is that you want to achieve. If you're doing skin resurfacing, you'd have to optimize the amount of time, the type of laser, and the type of the patient's skin as well, depending on the outcomes that you want. If you're doing photodynamic therapy for the treatment of cancer, then you have to optimize the type of photosensitizer that you're using. You have to optimize the light energy that you're using. What power output of laser are you using? What fluence are you using? And also you have to optimize many other things. If you're using it for photobiomodulation, you have to optimize also, if you're using red light, you'd have to optimize how deep is the wound, how wide is the wound. So there's a lot of optimization stages that you actually have to use before you decide on going through with the treatment procedure. That is why dermatologists are faced with a lot of time optimizing a therapy that would take them less than 20 minutes to operate, and they charge you $5,000, and you wonder that this therapy just took you 20 minutes to complete. Why are you charging me $5,000? Because the work involved in using light therapies is not just the delivery of the light and the actual work, but the optimization and everything else is very important as well. Okay. Which class of infections can you fight with this technique? This is a very good question. So the type of infections that you can fight with this technique, and we're talking about photodynamic therapy, not photobiomodulation. So when we're talking about photodynamic therapy for the treatment of infections, you are looking at having done an accurate diagnosis of the infection. The reason why I make it sound very important to do an accurate diagnosis is because the outcome of photodynamic therapy will depend on the photosensitizer that is available to absorb the photons of light that will cause a effect. That is why I mentioned about acne, that if you want to use light to treat acne, then you have to be sure that the person that you're treating has got the bacterium, Propionibacterium acnes in their acne lesions. Because if they don't have the bacterial cells in the acne, then you're going to use the red light and it won't do anything, because on its own, it won't really achieve anything other than finding the molecules inside the Propionibacterium acnes and then causing the phototoxicity. So there's the good thing about different types of bacteria is that most of them produce different types of molecules that are photoactivatable. So these are something that we call natural photosensitizers. Even plants have got these molecules as well. That is why even when we're treating cancer, we get these photosensitizers from plants. We can make them from different types of material, but the most important thing is that compound that has to be available for the light to work. So the question of what type of infections would benefit from treating using light, it would have to be infections that have bacteria that produce the particular type of molecule that would correspond with the particular type of light that will cause the phototoxicity.
Wonderful explanation. And while you're going to the next slide, some of your friends wanted the link to be shared through to your friends. They're unable to reach here, so it's on your WhatsApp. Just go on your WhatsApp.
I should.
So some of your friends just go on your WhatsApp. You see.
Okay, let me open it.
Yes. So you can share the link to your colleagues and friends who are waiting to get this link so they can click on it. So wonderful explanations till now. The healing effects of light, photo modulation, and photodynamic therapy, which so many people go on your WhatsApp.
Am I WhatsApp?
Yeah. The link is there. This link is there.
Oh, you sent me the link?
Yeah, you
okay,
have
cool.
the link.
All right.
So you can just share with your groups who are waiting to join this talk, and they are wanting to have this link so that they can.
Okay.
Can you see?
I just shared it.
Can you see?
Mm-hmm.
It's,
okay, cool.
Yeah. So yeah.
Yeah, looks like we getting a lot of questions. So let me just.
You can finish your slides and then come back.
I think these were the most important slides. Yeah. Let me see if I have to go further.
Just to let you know, we have scientists, we have biophysicists, we have astrophysicists in the group. They are students of public health. They are doctors. So you have a very wide audiences.
Oh, great!
From every perspective.
Yeah. So let me just finish off with the slide. I think this should be the last one. Then we can go back to the questions and focus on the questions. So light therapy is really a very very beautiful field of study in medicine, because apart from the things that we've spoken about, the other new development is in regenerative medicine. So in regenerative medicine, you're looking at a multidisciplinary science where we are using different types of resources, materials, and mechanisms to do really cool stuff like create or develop organs. I was just looking in the news that there was a guy that received a heart transplant from tissue regeneration. Now already we're living in 2025, and there's already people out there that are literally creating and producing actual organs that can be implanted in tissue. So regenerative medicine is a very new science, but this is a multidisciplinary field where we're using biomaterials, we use a lot of growth factors, and the most important thing is the stem cells. So stem cells are a type of cells in our bodies that are very young in their embryonic development, and they have not specialized into any cell. All the cells that we have in our bodies, our skin cells, our heart cells, our nervous tissue, all the cells were once in the form of a stem cell that through growth factors and DNA regulation and stuff like that, they became the organ that we have today. So stem cells are still present in our body in different parts of our organs. We still have stem cells. And in regenerative medicine, you're looking at isolating these stem cells, which are more abundant in younger people, especially babies. The older we grow, the less stem cell storage reserves we would have. But in regenerative medicine, what we are looking at is we use stem cells to actually produce different types of organs. And how do we do that? Remember, stem cells are undifferentiated cells, so they can really become any type of cell in the body as long as you expose them to the right growth factors and you give them the right blueprint of what cell they need to become. So I've got colleagues at the University of Johannesburg that are really doing very interesting work in programming stem cells to become a particular cell type. So this can be used in a lot of treatment procedures. So you can actually create a bone tissue from stem cells. You can create heart tissue from stem cells. You can create liver tissue. Let's say people that have liver damage and they need a liver transplant. Livers are not easy to get. We can literally get stem cells, program them into liver tissue, and transplant them into a person. And why are we talking about regenerative medicine and light in the same conversation? It really is because of the concept of photobiomodulation that I spoke about. Remember, photobiomodulation is when you're using light to stimulate tissue growth. So in regenerative medicine, this is one of the disciplines that are actively benefiting from photobiomodulation, because if you were to use the growth factors without actually photobiomodulating the cells, maybe it would work or it won't, but it would be a very slow process. So what light does in regenerative medicine is to actively photobiomodulate these cells, so that a process that could have normally taken, let's say, biologically using natural growth factors, a process that would have taken, let's say, 8 to 12 weeks, if you photobiomodulate that same process using photobiomodulatory activities of light, then you can achieve the same thing in much less amount of time. And this is really beneficial when it comes to regenerative medicine. So really, that's a growing field.
Somebody wanted to demonstrate on the hand the effect of the light. So if you can see the skin here, and like on my skin, can you explain to the audience what you mean by the regenerative? Like what part of the skin actually the light is affecting?
What part of the skin?
Right. Is it only the superficial, or the light can go in?
Okay.
Right.
Okay.
So if this is the skin,
if this is the skin, the different types of light, remember I keep repeating that the different types of light have got different penetration ability because of their different energies. So if you're looking at light from lower down the visible spectrum, let's say from blue light going upwards, those that light doesn't have a very deep penetration ability, but then it goes into the superficial surface of the skin. That is why blue light can be used for the treatment of jaundice, for instance. So when a baby has jaundice, we use blue light because blue light is energetic enough to just enter the superficial surface of the skin where the hyperbilirubinemia is. And how does blue light treat jaundice? It really is for two reasons. Bilirubin is a molecule that is yellow in color. So when bilirubin is exposed to blue light, what it does is it undergoes some form of photoisomerization. So remember, jaundice is because a baby cannot excrete the bilirubin that's accumulating in the bloodstream. That's why they become yellow. But then when we put them under a blue light lamp, what we are actually doing is, instead of waiting for the liver, the premature liver of this baby to conjugate all the bilirubin that's produced, we're actually using blue light to do the conjugation alongside the liver. So what the light does is that it changes the conformational structure of the bilirubin molecules, photoisomerizing it from its water insoluble part that the body cannot excrete to a conjugated isomer that the body can actually excrete. So that is why we use blue light for treatment of jaundice. The reason why we have to use red light for other deeper tissue procedures like wound healing is because when we go up the visible spectrum, remember we're going up but lower in ionizing radiation, but the penetration depth is increased the higher we go. That is why red light can penetrate deeper than blue light. And that is why red light is more likely able to photobiomodulate deeper tissues than blue light. And someone even spoke about infrared light as well. It really is because infrared light can actually penetrate deeper than visible spectrum. That is the reason why if you take a remote control and you press it on your arm, you can literally get your TV working because the infrared light can actually pass through tissue. That's how deep it can penetrate. So the penetration of different types of light really depends on the amount of the type of light that you're using, and of course the type of skin. Remember, skin is different, not just in color but in texture as well. So I'm a black person. I've got a lot of melanin in my skin, and Rajiv is lighter than me. That means I've got more melanin than Rajiv. So if we were to achieve the same thing using light, maybe Rajiv would have to get more irradiation time, would have to use something with more power output, whatever the optimization processes that we do. But the truth is, my response would not be the same as a person who's lighter than me. All right. So the outcomes, the biological effects of light will not only be dependent on one aspect, which is maybe the type of light that you use. You can use the same type of light on 5 different people and have 5 different outcomes. Why? Because the different types of skin tissue, the color, the texture would influence how deep the light goes. I was talking to a colleague of mine that are developing a light therapy procedure for the treatment of dementia and Parkinson's disease and stuff like that by causing photobiomodulatory effects of brain tissue. So this procedure is called transcranial infrared laser stimulation, TILS. So what it does is that they use some infrared light energy to expose to the frontal lobe of so that the light enters the brain and then it stimulates the physiology of the brain. So what it does is that it causes, remember we saw how light can actually cause the production of angiogenic factors. So what that does is that people with dementing disorders, usually one of the reasons is that they do not have enough blood supply into the region of the brain that needs the processing. So when you expose light to that part of the brain, what you're actually doing is you're causing a photobiomodulatory effect to the brain vascular system that actually causes some form of tissue regeneration. And we're talking about the blood vessels. So the blood vessels would actually increase. They increase in number, they increase in size, thereby increasing the perfusion of blood into the brain, and that's how it helps with dementing disorders like that. So that's just one of the examples that I would give in that regard. I think I will stop here, because if I go further, I'll just go into other details that may not be very relevant
And
for
this
this
is the
discussion.
importance, because as you mentioned, you will be breaking down your series into easily understandable topics.
Yeah.
And today you were wanting to give an overview. As a PhD, you can imagine how much is there to share with the audience.
By the way, I got hijacked yesterday, right?
Right.
Yeah, so I've made a deal with Dr. Rajiv. I'm going to prepare more in-depth presentations where I will break these things into bite-sized, digestible content. Here, it was just me trying to introduce what this is. But I think in the next, just an overview, and I did not do so much justice to it.
A lot of our students are also career driven, and they would want to listen to you about what are the career opportunities before they go to PhD, how they can reach that zone. And especially students from India and Africa who are logging in and listening to you, they are very interested in career advice in the future, how this can help in career. So so you can take some comments and questions, and then I'm sure we will be finishing this talk.
Yeah. So let's.
Can you see the comments? There's few comments in the last.
Yeah, I'm seeing a lot of. Can old age patients with wrinkled skin take this without fear of cancers?
So I think the question is like elders, the people who with sagging skin and wrinkled skin, they are going for various kind of therapies, and light therapy is one of them. But should they be scared of cancer? Can this lead to cancer?
So light in itself cannot lead to cancer. Cancer is a very complex pathological process. But how I would respond to that question is, if a person is predisposed to, let's say, a melanoma, let's say an older person is already predisposed to a melanoma, that is not growing or becoming a cancer because the body has got its inherent mechanism to stop a cancer from growing. Now, if by some unlucky situation a person like that gets to a dermatologist to remove wrinkles, and they decide to use red light or near infrared light for that matter, yes, most likely the light can actually propagate the development of cancer, because remember, cancer is the fast growth of cells. So if you are going to use light inappropriately, you are going to enhance the proliferation of the cancer. I've actually seen this in the lab. I would be experimenting on cells that I have not added the photosensitizer to. So if you use red light on cells that do not have a photosensitizer in it, you literally cause overproliferation of those cells. So yes, if there's an existing predisposition to cancer and for some unlucky reason it gets in the path of red light, yes, there will be an overgrowth of that cancer. But the light in itself would not at any point cause cancer. The part of the electromagnetic radiation that causes cancer is the ionizing radiation, which is the radiation that we spoke about from the UV region going down the spectrum, which is shorter wavelengths, higher energy light in that context. So visible light does not cause cancer. Infrared, microwaves. There's another conversation about the increased use of Wi-Fi. Does Wi-Fi cause cancer? Well, the science says no, but there's still more studies that we need to do to really answer that question. But the science of light, if you're going up the spectrum, that's low energy light that can cause photothermal effects, but they are not ionizing uh, radiation. So that's really the main difference. So the only part of the spectrum that can cause cancer is when you're going down the spectrum. That is why people even talk about sun exposure. They say don't expose yourself to the sun, you will get cancer. Well, there's there's a pro and con when it comes to sun exposure. Every person needs the right amount of sun exposure. If you do not expose yourself to the sun, you are going to deal with many other problems, because I gave an example of vitamin D. Your immune system depends on sun exposure. So, in as much as we avoid sun exposure, we should be sure that we give ourselves the right amount of exposure to the sun while we are avoiding overexposure. Too much of everything is poisonous. Even if you have too much water that you need right now, you it can kill you. Even oxygen is the very thing that we need to breathe, but too much of oxygen can also kill you. So that's really what we need to understand about light as well. Light is good for us, especially UV light is good for us, but we need it in the right amount of exposure. Biology and physiology usually works in a concept called homeotic response zones. So in homeosis, you're looking at something that you need at the right amount, over which can become toxic, below which would be irrelevant. So there's certain things in biology that you need to have at the right amount of exposure, right amount of even in biochemistry, that's the same thing. There's things that we need at the right amount of exposure and energy. So we should really be very careful how we handle information out there. I've heard a lot of people talk about don't go sunbasking, it's dangerous for you. No, that's wrong information. The sun is good for you. Just do it at the right exposure times. Too much of it really would cause some problems.
Thank you. I think that was wonderful explanation, and that brings us to the end of the telecast. But also, some of the comments and questions have given us the future topics.
Yes.
So light and cancer is a huge topic in itself. I'm sure at some stage you will be coming back with extra knowledge and more research-based answers. And in today's world, the knowledge is like expanding in every direction. It is very, very difficult to be very specific on specific questions. But for audience, I think they understand that here it's a communication, start of a process of communication where we have more topics coming in the front. May you have the last word before you close this telecast today.
Okay, so I will finish off by saying thanks for joining to begin with. And I'd just like to invite you to join us into our future conversations. This was a very random conversation that we had. We were just trying to introduce the topic, get you guys interested in the discussions that we're going to have in future. So I just really like to invite all of you to subscribe to the channel so that we get involved together. You can send us the questions, the comments. If you have suggestions on the topics that you want me to look into and maybe come with a more detailed topic on it, we will do that. And I just also want to mention that I've also started a podcast channel on YouTube, and I think it will be in the description link below. Please do subscribe as well. It helps to spread the word out there. So yeah, I'd just like to really invite every one of you guys to really join our conversation, subscribe to the channels, get involved, ask the questions, and give us your suggestions on the topics that you want us to talk about. Thank you.
If you want to be a speaker, you can reach out to yourself on the topic.
I actually have a colleague of mine that I've seen comment on
He's made the comment, right?
and I'm sure he would want to. He's doing some really interesting work using nanoparticles and stuff like that. Yes.
Yes.
Yeah. So I think we'll get this colleague of mine to do a presentation as well, because he's currently working on some really cool stuff in photobiomodulation, photodynamic therapies. So I might engage with him to see how we can take different parts of the conversation together.
Maybe Evlin can finish it by telling how people can get involved. Evlin, you can.
Yeah, absolutely.
Yeah, let me give you the.
Yeah, so thank you everyone for coming in and joining us with this. There are a lot of ways to reach out to us in the comments, through email, over Facebook, LinkedIn, just DM us, Instagram, just reach out to us. We have that information on our page and also at the end of most of our live streams. So please reach out to us and share your comments, questions, concerns, and especially if you're interested in joining us and possibly giving us a presentation of your own and joining us on our speaker series. So thank you so much. And thank you to Dr. Elvin as well. And bye.